Please be seated. As chair of the Whittier College faculty and on behalf of its faculty, I welcome you this afternoon to this august occasion. We are quite privileged to have a number of guests and guest groups with us this afternoon, and I especially want to recognize some of the high school communities that are joining us this afternoon, and we welcome you to the Whittier College campus. In addition, of course, there are the great students from Whittier College represented here in our audience, as well as the faculty and trustees of this institution. A special welcome, however, is due to Mr. Yuki Hayashi, and let me recognize Yuki here today. It is not only is he the father of Whittier senior Maki Osata, who is joining us. There she is, yes. Not only is he her father, he is also the one who has in many ways helped to make this special occasion possible for us today. And of course, we welcome our esteemed guest, Nobel laureate Luc Montagnier. Our Quaker forebears established the college with a mission to promote tolerance, internationalism, service to others, respect for the diversity of opinions, and reverence for the value of listening to others. It is my impression, Professor Montagnier, that these habits of mind and behavior are often the key ingredients to the kinds of breakthrough in science for which you are so acclaimed. We are indeed honored by your presence today. And as we begin our ceremony this afternoon, we acknowledge the Quaker roots of this institution by following the tradition to observe a moment of silence. Please join me in this time of communing together in silence. So be it. Thank you.
Good afternoon. I am Charlotte Borst, Vice President for Academic Affairs and Dean of the Faculty here at Whittier College. First, I want to thank those who helped with the logistics and ceremonial aspects that made today possible. In particular, I want to thank Russ Litchfield for playing the pre- and postlude. Joining me on stage, in addition to Professor Montagnier and Professor Joe Price, are Whittier College President Sharon Hertzberger, three talented students, Joshua Smith, Vanessa Parada, and Apollonia Galvin, Fletcher Jones Professor of Molecular Genetics Dave Borghese, who is Director of our new Center for Health Science and Policy, and Professor of Chemistry Devin Emoto, who for several years now has taught a multidisciplinary course on AIDS, its prevention, and its treatment. At Whittier College, we have no higher recognition than the awarding of an honorary doctorate. We make such awards with great pride to those whose lives have personified the convictions, the ambitions, the values, and the accomplishments that we celebrate here at Whittier. Today, we add to the awards of someone who has already received what is widely regarded as the highest recognition in the world, a Nobel Prize. President Hertzberger, will you please come forward, and will Professor of Chemistry Devin Emoto please escort Luc Montagnier to to the podium and present him with this award.
Professor Luc Montagnier, your work saved lives. For your discovery of HIV, the virus that causes AIDS, you were awarded the 2008 Nobel Prize in Physiology or Medicine. Your isolation of HIV subsequently led to the development of a diagnostic test for the virus. This has allowed people to know if they are infected and to take measures to avoid infecting other people. Furthermore, your work showing that HIV infects helper T4 cells has paved the way for people to understand how HIV replicates, and as a consequence led to the development of antiretroviral drugs that are used to treat people with AIDS. These drugs have kept people alive much longer and with an improved quality of life. And even before your work with HIV, you made significant contributions to the field of understanding how retroviruses cause cancer and to understanding the general function of the immune system. For your contributions to science, you've been awarded numerous honors in addition to the Nobel Prize in Physiology or Medicine. These honors include the highest honor that France awards, which is the Légion d'honneur in 2009 at the rank of Grand Officer. In addition to that, you were offered or you were honored with the Lasker Award and the Gardner Award, awards and honors given to scientists that have made significant contributions to the field of biomedical science. You are currently the president of the World Foundation for Medical Research and Prevention, and your ongoing endeavors at the Institut Pasteur in Paris are aimed at finding a vaccine for AIDS. We are hopeful that you will be successful in this endeavor, and you have stated, you have been quoted, it is clear that prevention will never be sufficient. That's why we need a vaccine to be safe. So for your tireless efforts to improve the human condition and for your brilliant work on AIDS, Whittier College is proud to host your visit to our campus today and to learn from you about HIV/AIDS research, past and present. Madam President, I am honored to present Professor Luc Montagnier for the degree of Doctor of Humane Letters honoris causa.
Luc Montagnier, upon the recommendation of the faculty and by the authority vested in me by the Whittier College Board of Trustees and the State of California, I hereby confer upon you the degree of Doctor of Humane Letters honoris causa. Congratulations. This will be your hood. Should we put it on you as we're not dressing up in regalia, but you'll get to see what it would look like should we do so. So, there we are. And should we take that too?
Thank you very much. I'm very honored and moved by this honor here. I'm very pleased to be here, and good afternoon to all of you. I'll be speaking about AIDS, but before that, I'd like to remind you we are human beings, and in one way we are like all the life on this planet. We are inheriting genetic memory for a very long time, 3.5 billion of years, probably many inventions from life. But we have also, more recently, a new memory, which is the cultural memory, transmission of knowledge from one generation to another. And this is what this college is doing, actually. It's very important. We are not human beings if we don't have this second memory. it's very important also to remind, unfortunately, that we are biological beings, very complex, and we are suffering of disease. So my task was try to contribute to the knowledge of the disease which are hitting much the world now. You have a slide showing that many infectious diseases are causing the death of many people in this world. And despite the research, the biological research, the medical research for now many decades, we are still fighting without much success. HIV/AIDS, malaria, tuberculosis, cancer. So I will focus today as a model on something I know best, is HIV/AIDS, and show you, excuse me, okay. But just reminding you that our genetic memory is not completely known. Of course, we have sequence. We know the sequence of all the human genome, but the surprise is that only 3% of this DNA is coding for proteins, they are really genes. And besides that, there are retroelements, including endogenous retroviruses, the same family of the AIDS virus, but they are in our genome for millions of years. Some retroviruses are probably good for us, but some are bad, and probably some of them are programming of genetic of death after some time. There are also related elements, retrotransposons, which can move genes, and cancer is not only due to mutation, single mutation, but also to the changing place of some of our genes. We should not forget about that. So we don't know too much about those structures, those elements, and they are also made of many repeats. Some are short, like the sign, some are longer, like the line, and they make up almost 50% of our genome. So we have still to learn a long knowledge about what is the role of those structures, because they are there, they don't change too much in millions of years. And of course, those elements, the retroelements and retroviruses, have a specific enzyme called reverse transcriptase, which have the property to translate DNA into RNA. That is, RNA then goes back to DNA. This is the main effect of this polymerase, RNA to DNA. And that DNA will integrate into our sequence itself. So when we started at the Pasteur Institute in 1983, our research on the possible role of retroviruses in AIDS, we, of course, took advantage of the knowledge of many of our colleagues before who worked on animal retroviruses and set up the stage for the knowledge of this new kind of virus. And we used the reverse transcriptase. My collaborator, Françoise Barré-Sinoussi, used the reverse transcriptase enzyme to detect what became later the AIDS virus. So this was summarized in a paper on September 1983. And of course, at that time, we were just very few people believing in this virus. We were about a team of 10 people in my laboratory, with some other people coming from a hospital in Paris. They are listed here. And we had good arguments to think this virus was the cause of AIDS. But nobody believed in us. And you have to remember that when we do new discovery, sometimes it's not accepted by the scientific community. And so it's not only good to find things, but we have to convince colleagues that we are right. And this takes some time, because at that time there was not many retroviruses known in men, only one discovered by our colleague Robert Gallo in the United States. And Robert Gallo was thinking at the time that the AIDS virus could be just a variant of this own virus. But this was not true. Our virus was very different, and it took us about 1 year to convince him that we were right and confirm, of course, the extent of our research. So this summarized the milestone of research of AIDS. Before, of course, was the identification of the disease itself in the USA. The first patients were diagnosed by Dr. L.H.D. in Los Angeles. They were young homosexuals suffering of pneumocystis carinii infection, lung infections. So after the first isolation of HIV and confirmation, then came, as my colleague knew, that this led to the prevention of AIDS transmission by a blood test, which was both elaborated in the United States and in our team at the Pasteur Institute. So this could eliminate the transmission of HIV by blood transfusion. Of course, not eliminating everything, because AIDS is, as you know, is transmitted also sexually transmitted, and of course it's very difficult to control the behavior of people. And AIDS is, of course, a medical problem. It's also a social problem and behavioral problem. We should not forget about this. In 1986, we isolated another virus close to it. It was the first one in West African patients. But we realized before that AIDS was not only a disease of homosexual men with many partners, not a disease only of drug abusers, but also, especially in the South, in Africa, a very large population disease, heterosexually transmitted as well. And this is very important to remind that not only AIDS, the HIV infection, it's some so-called high-risk people, but also people which have no risk factor, especially in Africa, at least not known risk factor. But we have to explain this difference. Why in the north only some high-risk people are catching easily the virus, while in the south many people can be infected and die of AIDS. So this is still a problem we have to solve. And of course, I won't spend too much time in my talk, because I will leave also the floor. I will be glad to answer your questions. After that, the important step was discovery of inhibitors. Of course, knowing the enemies, it's easy to think of inhibitors, but it took some time before we could get good viral inhibitors of viral multiplication. And their combination was successful only in 1996. So between 1981 and 1986, many people have been dying of AIDS. Meanwhile, of course, the question was also raised how the virus can induce the strong depression of the immune system which characterizes AIDS. Some people with AIDS have almost no T lymphocytes. Lymphocytes used to kill infected cells. There are almost nothing left. And therefore, the first idea was maybe the virus killed the infected cells. It does, but it does that sometime late in the infection, not at the beginning. It takes several years to develop. And probably there are some indirect mechanisms which explain the disappearance of the immune system. I will come back to that. So it's not a simple disease, not a simple infectious disease. It's a very complex disease, and actually it's the model of many chronic infections. Some of them are not known to be, we don't know the infectious factor, but it has to be discovered. So I don't want to go too much into detail, but just to say that we know a lot on this virus, on its genome, on the DNA. We know a lot of those genes, of its genes, but we still don't know everything. And still there are some important research. We could lead some new treatments, a new vaccine, if we know better all the form of this virus, which is not completely known. Just to remind you that, of course, we have now enhanced, thanks to the research of many laboratories and also the pharmaceutical industry, we have access to many good inhibitors which act on different steps of the virus. The reverse transcription, RNA to DNA, the production of viral particles by a protease cleavage of some viral proteins, protease inhibitors, but now there are also fusion inhibitors, fusion of the virus to the cellular membrane, and also inhibitors of the binding of the virus to receptors. But it has to be stressed very strongly that none of those combinations cure infected people. It's not a cure. There is no cure. Because there is a reservoir of virus, some form of the virus, which escape the treatment. And this treatment has to be given for life, with some side effects, tolerance mutation, resistance mutation. And if we stop this treatment, the virus comes back. It comes back from something already existing in our body. So we have to find out. So this slide, which is very classical, shows the timeline of the virus infection. Good multiplication of the virus at the beginning, then some immune reaction which will decrease the virus production in red, and at the same time the immune system begins to decline. And at the end, in general, between 5, 10, 15 years, there is almost no immune system left, at least the cell-mediated immunity, and people die of opportunistic infection and also of cancer sometimes. Except a few people. Few people are not following that model in that they are reacting very strongly against the virus, so they keep the virus down to a very low level and are not suffering from immune depression. They are called L.H.D controllers or long-term survivors. There are a few people, that maybe between 1 and 5% of the infected population. And those are models because they teach us that the immune system itself could control the virus infection. So we have to find out how. And if we learn how to do that, we may extend this knowledge to many people. If now we treat by the combination of viral inhibitors, we are more in this kind of curves in that we decrease very much the production of virus. We don't detect any more viral particles in the blood. Still, we have some other virus probably in lymph nodes or in other tissues remaining. And we see a partial restoration of the immune system so that people don't die too much of opportunistic infection. Sometimes they still die, but most of people can recover and work normally. So this is why now the life of many millions of HIV-infected children can be preserved. This happened, of course, first in developed countries, but now it's expanding slowly to patients of developing countries. Another characteristic of this virus is its high potential to change all the time. I think it's part of the strategy to resist the immune reaction. It changes, so it exposes more variable part of its surface protein, and then, of course, by one few mutations, it could escape the reaction. And there are many mechanisms. I don't want also to go into much technical detail, just to remind you that one of the factors of variability is oxidative stress. What is oxidative stress? It's an imbalance between molecule very reactive, derived from oxygen, reacting with DNA, proteins, lipids, carbohydrates, and of course damaging those compounds. But especially DNA, they can induce mutation. And these mechanisms also exist in cancer. So we should not forget that there are many analogy, similarities between HIV infection and cancer in that both have a very high variability potential. Tumor cells vary all the time. HIV also does. And partly also because both are caused by oxidative stress. So if we can neutralize oxidative stress, we can prevent some risk of having cancer, and we can also prevent the variability of infected. We found some time ago, in the early 90s, that actually many cells, many lymphocytes, many blood cells were dying by a process called apoptosis, which is some kind of suicide, due to the HIV infection. But this was not caused by the virus itself, because many subsets of that population were not sensitive to virus infection, but still they were prone to die. So as underlying the importance of indirect mechanisms for decreasing the immune reaction, the immune system. Another finding which was also striking is the fact that our immune system normally is programmed to decline. We have a good organ at birth called the thymus, which controls the programming of lymphocytes, T lymphocytes. And there are many lymphocytes passing through this organ at birth, but this organ is slowly involuting with age, and we are about 17, there is almost nothing left. And the surprise that an AIDS patient, only 25-year-old, had the same involuted thymus that a non-infected person, 70-year-old, indicating also an indirect mechanism of evolution of the thymus induced by the virus infection. But partly reversible if we treat 25-year-old AIDS patients by viral inhibitors, partly restoration of this thymus function. Oxidative stress, I mentioned it already, and there is strong evidence that oxidative stress by itself can weaken the immune system. It's a little too complicated, I will pass through this. But there are evidence of oxidative stress in HIV infection, even before the decline of the immune system, and this oxidative stress actually probably contributes to the decline of the immune system, oxidizing lipids, LDL in the blood, oxidizing proteins, and oxidizing DNA, inducing mutation of inflammation. So we have to control this as well. This is not always done. It is known that some of the treatment, especially the reverse transcriptase inhibitors, are also inducing oxidative stress. So doctors have to counter this by giving good antioxidants, including glutathione and some of the products made by our colleague Dr. Yuki Hayashi, the fermented papaya preparation, which is also a good antioxidant. Actually, I met Yuki in 1994 at the Yokohama AIDS conference, and at that time we didn't have this combination of strong inhibitors of virus infection, and he proposed to me to try FPP, and we had some interesting results at that time. Now, what is the origin of oxidative stress? You could ask, is it due to the virus itself, or is it due to some co-infection? Probably both. And there is a protein which is made by the virus called TAT, which has many functions, but including decreasing expression of one of the key enzymes for fighting oxidative stress called superoxide dismutase, or SOD. So part of the oxidative stress may be caused by some viral protein, the TAT protein, the surface proteins, but probably also co-infections are very important. And we also tend to forget about this. AIDS is caused by a virus, no doubt. Well, no doubt for many people, few of our colleagues are against this idea, but I think there is overwhelming evidence that HIV is causing AIDS, but it's probably helped also by some cofactors, infectious cofactors, and also the general environment or behavior of the person infected. And so when we treat by antiretroviral therapy, we are still left with something behind. And this is shown by the fact, for instance, we can still detect infectious particles from the blood. So people which are well treated, well responding, could be still infectious. We should not forget about this. And the restoration of the immune system is not complete. There is still some anomalies, biological anomaly of the immune system, so that we have to find out the role of the remaining virus, called the viral reservoir. So, after some time spent in Africa, I realized that, of course, the policy now of helping developing countries by making them available at low price viral inhibitors was very good, but not sufficient. Necessary, but not sufficient. Because it's impossible to keep that policy for a very long time in countries in which you have 5-10% of people, of the total population, people infected with HIV. All the budget of the country, and even in a developed country, if we had, for instance, here in the United States, 10% of people infected, it would be impossible by any health policy to keep them on their antiretroviral therapy for a long time. So my proposal has been to find some complementary treatments which could be added after a short-term treatment by antiretrovirals, so that this treatment will be short, could be affordable, and will not have too much of the side effect. So this is what I'm saying now. But it's easy to say, but not easy to do. So how can we do that? Well, the first idea, of course, is to boost the immune system, because we have also evidence of some people which are naturally fighting well the virus, controlling the viral immune system. Can we don't do it also after a short-term treatment by antiretroviral therapy, where the immune system is partly restored for the infected persons? So the idea was to set up a vaccine, a therapeutic vaccine, a vaccine which be given not to people for prevent infection, but to help curing the infection in people already infected. And knowing also oxidative stress play an important role in decreasing the immune reaction, gives some good antioxidants. And of course, finally, would be to identify and target the viral reservoir. How can we do that? Well, this cartoon shows what's happening when we treat by antiretroviral therapy. We reduce the virus to almost nothing, but still a reservoir is still left. And when we stop the treatment, this reservoir gives back the virus multiplication to a very high level. So the idea was to add, after the first restoration by the treatment, some protein of the virus to immunize, to reinforce the immunization against the viral proteins, especially the proteins which are at the surface of the virus, which help the virus binding to the cell membrane. And to be short, this also depends on the structural identification of the three-dimensional form of the viral protein, of the binding to the receptor. And there is especially a loop of the surface protein called the Whittier loop, which binds one of the co-receptors to the virus. But unfortunately, this loop, many of its parts are highly changing all the time by mutation, so the virus can escape very easily the antibodies or the cell-mediated immunity raised against this variable part. But there is only a small part, a crown here shown in green, which is conserved because this part binds the co-receptor, so the virus cannot change, otherwise it will not bind the cell membrane. So the idea was to try to keep this crown and to raise the antibody, the immune response against this crown, instead of raising the immunity against the variable part, which is shown here in black. So what would be the result of this modification? We can do it by molecular biology, of course. Well, it's first to treat by antiretrovirals, and then treat by the vaccine. Normally, when we stop the treatment, the virus comes back, but if you vaccinate, the hope is that the virus will not come back, no rebound, and we will keep the virus down for some time. And if the virus comes back again, we can vaccinate again. So this could help. The problem is to show this is working. We have some evidence in animals, but of course making clinical trial with that preparation takes some time, so we are not reached that stage yet. But this is the idea, to use, after reducing the viral load by treatment, use antioxidant and this vaccine, which could be made also of other protein as well. Well, is that sufficient? Probably not. And we need also to target what remains in terms of the virus. And we have been working now on the nature of the viral reservoir and the question mark, what is it? It's probably some viral DNA. But to show that, I have to show you some very esoteric, perhaps esoteric studies which show actually a new property of some viral DNA, some sequences of DNA from viruses and bacteria. This is the production of waves. And here we are entering a new field, which has to do with physics. So this is also a message I like to convey to you, to the young generation. It's now, discovery can be made by interaction between disciplines, translational research. And now we need also the contribution of physics. Physics have shown us that in the universe, there are not only matters, but there are also waves. And some particles, as you know, this is the ondulatory mechanics, have both the property of corpuscles and waves. And I think this notion should go now to biology. And I'd like to show you an example. This is just the beginning, of course. We have many unknown, but still we find by experimental research some very strange effects, some very strange properties of some of the DNA sequence, which is the emission of low-frequency electromagnetic waves. It's a resonance phenomenon and used by a still lower frequency of electromagnetic waves. So in order to capture those electromagnetic signals, we use a device, not done by us, but done some previous researcher in France, in which we can detect the waves produced by a dilution of DNA in water, high dilution in water, amplify it, and analyze it in a software, in a laptop computer. And so in order to detect those signals, we have to dilute out and filtrate the DNA solution and excite by some very low frequency waves, less than 100 Hertz. And if we are lucky, we find resonance phenomenon emission by some of the DNA dilution, high dilution, of frequency which are a little higher, and the amplitude also is larger than the exciting system. This is shown, for instance, by here by a transform Fourier. So this is done due to the French mathematician Fourier analysis, which can analyze a complex waves by units of frequency. And this slide shows that a range of frequency from 0 to 20,000 Hertz, and in yellow is a very low frequency for the part of the exciting signals. This is the background noise. But if you have a signal, positive signal coming from DNA, you get a shift to higher frequency in blue. And so this is easy to detect, but now we have also more refined system to quantitate this response. And the surprise is that many bacteria which are causing disease in men have DNA sequence emitting this kind of signals, as well as some viruses like HIV, hepatitis B, C. But also this can be detected in the blood, in the plasma of humans infected with the same bacteria or viruses. So we are now going from fundamental research to medical applications. And of course, we have to work a lot to show the role of dilution in water. Some dilution works, some do not, and this can be explained by this simple, probably wrong explanation that we are just beginning. So the idea is that DNA induce in water some structure which at low dilution are bound to themselves. They form a network, crystal-like structure, which does not vibrate. Only when we dilute sufficiently the dilutions, we see the free vibrating polymers induced by the DNA in water. Okay? Okay. So, bacteria are showing that. Not all the bacteria. So far, only bacteria which may have a pathogenic potential. If you use probiotics bacteria, nothing happens. So maybe this is a way to distinguish between good and bad bacteria in our gut. And so for some time, we have been trying, of course, to show what kind of DNA sequence could be involved. This is a long-term work. We have started, but we haven't much results. We can, in a few bacteria, I don't want to go into the detail, we have identified the gene, but just a few of them. And the structure produced in water, of course, are totally resistant to anything, to any enzyme, because they are just water. And this is important because those structures, which are matter structure, can have some, by themselves, some effect on the immune system. So we are dealing now with something completely invisible to the immune system that may also contribute to the immune deficiency in HIV infection. So we have published this in a journal which, in fact, bridge physics and biology, interdisciplinary science, computing, life science. And we have been focusing mostly our work on few infectious diseases, starting with HIV, but also some other infectious diseases as well. So the surprise came when we studied HIV-infected patients. Actually, we found that the signals are not produced by viral particles, are not produced by RNA, but they are produced by the proviral DNA, which is made by the reverse transcription. And we check, of course, with pure infectious viral DNA. But the surprise that in the case of patients, only the plasma, the patient treated by antiretroviral therapy and doing very well, having no detectable viral particle in their blood, they were emitting signals. And we showed that the signals were really coming from some HIV genes, DNA genes, but also not only the plasma, but also the red blood cell fraction. Red blood cells have no DNA. The mature blood cells in our blood have no DNA at all, but probably some viral DNA is associated with the red blood cells. By some way we have to find out. So just to show you, to illustrate this is the case of patient in which we have worked in Ivory Coast, in Abidjan, in our center, created by our foundation in Ivory Coast. And we show that at some dilution, we have this kind of shift of signal and also increase in amplitude in patients treated by ARV. And this slide shows you some representation of the signals. On the left, you have the noise. On the right, you have a positive response. First, on the top, the crude signal of it is recorded. And after, on the middle and the back, on the bottom, you have the Fourier transform analysis by different software. We also, of course, were eager to see which kind of viral DNA could be present in the plasma of treated patients. So this table, which is a little complicated, I don't want to go into the details, we are comparing On the top patient, we have been not treated and having no sign of AIDS-like disease. In the middle, people which were already having AIDS sign but were treated by antiretroviral therapy. And in the bottom, patients which were also sick but not yet treated. And the outcome was only the patients treated by antiretroviral, having no more virus load in their blood, showed the signals in their plasma, in the red blood cell, but also we had evidence of that some genes of HIV were producing the signal. This is shown on this slide by the technique we call PCR, polymerase chain reaction. We have modified it to make it highly sensitive, and we show also that the plasma of patients have the same gene producing signals that the pure HIV DNA. So what are the conclusions from this work? Of course, this is a new technology, and we have to refine this work by also talking and having some collaborative work with physicists. It's very important to give the basic explanation of this, but also there are some medical applications, because now whatever the situation of that DNA could be used as a biomarkers. Now, after antiretroviral treatment, we have no biomarker because the viral load, the viral RNA detected in the blood is almost zero. But now we have a marker which resists this treatment and which could be used for other kinds of therapeutics aimed at eradicating this form of the viral reservoir. And we have two techniques, the signals, which is rather short, and we have the PCR, the nested PCR also, which can be used for this. And we detect this DNA in the blood and maybe other fluid, other bloody fluids. So it's important to remind you that people which are treated by antiretroviral therapy are still infectious, may be still infectious. Of course, we can argue that we are detecting DNA fragments in the plasma. But previous work show also that in the red blood cell fraction, we could detect infectious virus. So it's quite possible that we are dealing really with infectious DNA, complete DNA in the blood. So how to explain this? Why the treatment pushes the virus to this kind of accumulation of viral DNA? And my hypothesis, so we are not dealing now with facts, but hypothesis, is that HIV DNA may be a consequence itself of the treatment. Because the treatment will completely suppress the normal way of multiplication or application of the virus, RNA, DNA, RNA. So we stop this replication and we oblige the virus to just use another way, more classical, which is DNA. But in that case, of course, this cannot be done by the viral enzyme, the reverse transcriptase. It has to be done probably by cellular DNA polymerase. So our research now is to find out which DNA polymerase can be able to do that work. So this summarizes what we are now: polytic cycle, RNA, reverse transcription, DNA, expression of DNA, RNA, and proteins. Here we are just a way to maintain the virus, no expression of the DNA, especially if this DNA is episomal, non-integrated into the cellular DNA, it will replicate by itself, but not being expressed into protein. So this is some way for the virus to remain invisible, but to keep its information until better days come to it. The better days for the virus when we stop the antiretroviral therapy. So whatever the function of that DNA, we still have a biomarker which allows us to look for complementary treatments which will eradicate the virus for good. So I think it's not really a dream. is something we can envisage for the next coming years. Maybe we will not have a vaccine, but we will have a treatment which will eradicate the virus infection, keep the virus to a so low level that it will not harm the patient's immune system at all. And so we are now testing some products, some compounds, some additional treatment. It's not to replace the antiretroviral therapy, but just to allow a short-term treatment by antiretrovirals, and then giving some natural products which could perhaps eradicate this viral reservoir. So this is the aim of my foundation. Of course, we are now collaborating with many other institutions, governments, to launch clinical trials, especially in Africa, in which the aim is to eradicate virus infection. I'd like, of course, to thank all my colleagues, especially those in Africa, from the Ivory Coast, from Yaoundé, Cameroon, and my colleague also from the World Foundation, and also from the company which is the spin-off of that foundation, called Nanectis. Thank you very much.
Professor Montagnier has graciously agreed to take a few questions from the audience. So if you have a question, can you please go to an aisle, and I believe that there are microphones being passed around, and so can you speak into a microphone so Professor Montagnier can hear your question? So again, go to the aisle and go to a microphone and then ask your question.
So is the reason why we don't have a vaccine, preventional vaccine, because we don't know all the forms of the virus?
Well, there are several reasons. One is the highly variable potential of the virus, and most of the candidate vaccines we have been using are targeted to the normal, naturally occurring surface protein of the virus, so that the immune reaction induced by the vaccine is mostly directed toward the most variable part of the virus. So, of course, the idea is to redirect the immunization against the most conserved part, but this involves molecular manipulation of the protein, and not on the candidate vaccine has done that yet. But the second reason, as you say, is probably the fact that there are invisible forms of the virus which could resist. There may be also some vectors of the virus in HIV transmission that have not been identified yet. So there are still a lot of research to do for that.
Since erythrocytes only live for about 120 days and circulate, and their production is mostly in the bone marrow, would there be some novel therapy, some type of bone marrow suppression therapy, similar to that of cancer chemotherapy, that might be used as an adjuvant to therapy to try to wipe out the reservoir stage?
Yes, it's possible that the DNA integrates or associated with the precursor of red blood cells in the blood, so the bone marrow precursor. But in that case, we should target the therapeutics against the precursors. So this is the opposite way you are proposing. You are proposing to use the bone marrow for complementary treatment. But if the bone marrow cells are already the virus DNA, I think the best is to not to use them. Of course, is to try to eradicate the virus in those bone marrow cells, which probably will be very difficult because they are multiplying.
Electromagnetic way of detecting HIV that you just talked about. How sensitive is it compared to PCR?
Yes, I think the correlation is very good between the two technologies. So the electromagnetic signaling is much quicker, detection is much quicker. It takes about 1 hour. PCR takes a little more. But in both cases now we have to extract DNA. We can also use fresh plasma, but if we freeze the plasma, we lose the electromagnetic signal production. But if we extract the DNA from the frozen plasma, we can still detect the signals. But DNA extraction takes more time, but it could be done in half a day.
So I have a two-part question, and it's more or less based on methodology, I guess. So my first question is basically, why is it your serial dilution levels at your lower volumes are not giving an EMS signal? I think most people that when they do serial dilutions, they think the higher the concentration, the more signal that's going to be arranged. But is there an explanation to that?
Yes, well, I gave one explanation in my cartoon showing that at low dilution, there are too many structural nanostructures forming in water, so they cannot vibrate, because it is a resonance phenomenon. And
there's still like an interaction between the two preventing that.
Yes.
Okay, and my next question has to do more or less, have you been able to correlate specific EMS signals to unique DNA particles that have been found? Or is it just a peak that's been given for EMS as a detection for the viral DNA?
I believe there are two kinds of signals produced by the nanostructure induced by DNA. I think probably it's a general phenomenon, but we are just detecting the tip of the iceberg. The most producing, the sequence which are the most intense signals producing. But in those signals, there are two parts. One which are similar for all the structure producing the signals, and more specific also behind this. Of course, they are more difficult to detect, but we have some experimental evidence that is indeed we can have specific signals induced in water for some DNA sequences. So there are some hopes to have, in a second step, a very specific diagnostic of that kind of DNA.
Shifting gears a little bit, you note the importance of interdisciplinary work with physics. What do you see the role of the social sciences?
Of course, it's a very complex disease, and we know that even though we have, for some other diseases, a good medical way to treat or to prevent, they are not completely used. And we have seen about 1 billion of people are infected with the active tuberculosis, despite we have good antibiotic treatment. So there are, of course, many other factors than simply that science cannot completely solve. And again, for HIV/AIDS, why this so huge difference between the North and the South? And my explanation probably, not only in different sexual behavior, I don't believe in that. I think it has to be due to the difference in the number of infections. If you have too many infections, you are stimulating the immune system, you kill your immune system actually. So people are becoming more fragile if they are infected by malaria, tuberculosis, parasitosis, and HIV. So the social role is very important. If we increase the standard of life of those people, like having access to sterile water, trying to induce some good hygiene habits, and fighting the co-infection, we can still probably do more than simply by a vaccine. I think even though we had a vaccine, it will not solve completely the problem. We have vaccine against hepatitis B. Of course, vaccine has been good at eradicating smallpox, almost polio. But of course, when you have complex disease, which are chronic disease, much more difficult to prevent this by a vaccine. So if we increase the standard of life of people of developing countries, we can achieve the same goal. And of course, prevention, education is very important. We can do it in school, but there are also some cultural factors which are against this. For instance, in Africa, there are still people believing in the role of God, of witch doctors and so on, in causing the deaths of people affected by HIV. So those people are not accessible to the treatments we are proposing. So there are many cultural factors. I do believe this will change with time, but it will take more time. And so I think there is some kind of a race between these epidemics, which is still very prevalent in African countries, some Asian countries as well, and the solutions, which could be both medical and social solutions, economy also.
Thank you very much. This question actually follows a little bit on some of the aspects of the answer that you just gave to the preceding question. And I also have to admit that this is a jointly crafted question from myself and another colleague. And we were very intrigued by multiple dimensions of what you brought to us today. And our question that we came up with was, in a complex global environment such as this, in which you are providing very significant leadership for the global effort to combat AIDS, we're wondering about your thoughts about how the financial resources can be most effectively mobilized to be used in a way that most effectively and efficiently addresses the needs of the populations most at risk and affected.
Well, complex question, complex answer. It's a global problem, and of course, I will not argue that the global fund, the international help, financial help, has been positive in saving the lives of millions of people by implementing low-price antiretroviral treatment. But again, it's not a long-term solution, because as many people, more people will be treated, infected, they will still transmit the virus. So the epidemic will continue, and at some point they will stop the treatment for any reason, too expensive, intolerance, and so on. So the only solution I see at the level of medical solution is to have some complementary treatment which will make people to stop their treatment at some point, not after a very long treatment by antiretroviral inhibitors. But I think to show this, the efficacy of this, we have to do some clinical trials with some compounds, some products which we think will stop the continuation of the viral infection. But we have to wait until the clinical trial gives the solution before we can really make a turn in the international policy against HIV infection now. Is that answering your question? Probably not too much, but we don't have magic solution. We need time.
In your approach to therapy, I was intrigued by the idea of the antiviral therapy followed by complementary therapy. And the question I have is, do you see this as being applicable to other chronic viral infections? And I'm thinking specifically of hepatitis C, which, as you know, affects about 2% of the American population. It's our number one reason for transplantation. And we've had similar problems in terms of vaccine development and also in maintaining sustained response. We can do okay with some of the genotypes, but not with the most common genotypes. And I find this a very interesting approach that potentially could be used in others. I was just wondered if you thought that was going to be applicable.
Yes, I agree completely. We haven't done any work on hepatitis C itself, but we are considering the application of this, what we are learned from AIDS, to other infectious chronic diseases, and not only hepatitis C, but also some diseases in which there are autoimmune components, like rheumatoid arthritis. We have shown the evidence of bacterial signal coming from bacterial infection in this disease, as well as some case of Alzheimer, Parkinson, MS, multiple sclerosis. So this model could apply to a certain number of chronic diseases.
We'll take one more question.
Okay. Hi. I have a non-scientific question, actually. So I understand that you are a co-founder of AIDS, and you've handled the situation with Gallo graciously. So my question is, what kind of outlook do you have on life, and what kind of personality do you think you have been able to do the things that you've done?
Well, my personality is more defined by my social interaction by others, so it's difficult to define myself. Well, if I'm to define, I can define myself as an explorer, Of course, we cannot find new continents like Christopher Columbus, but we can find new continents still in the universe, even very close to us, in our body. There are still many mysteries. How can we start from one single cell and become an individual, a very complex organism? That's something which is fascinating me. So I think also, well, my clue in my research now is to think that waves, electromagnetic waves could play a large role, not only in disease, but also in normal functioning of our body and our brain also. So this is how I can define myself. I try to find new islands, new continents in my body.
All right, we have a few Whittier College students who would like to present a gift on behalf of the college to Professor Montagnier. So would they step forward, please?
Good afternoon. I will now say a few words in Dr. Montagnier's native tongue. Bienvenue, c'est professeur Montagnier. Je m'appelle Apollonie Gavin. Je suis étudiant en deuxième année de licence de français et d'anglais. Tout d'abord, je tiens à vous remercier de la part de tous les étudiants de Whittier College d'être venus sur notre campus. C'est un immense honneur de recevoir un scientifique comme vous qui a tant fait avancer la recherche sur le sida. Vos découvertes dans ce domaine ont changé et continuent de changer la vie de nombreux jeunes de notre génération. Et nous vous en remercions tous. Je laisse maintenant la parole à Joshua et à Vanessa, deux étudiants de biochimie.
Merci beaucoup.
So I would like to start off by expressing what an honor it is to follow a Nobel Prize winning professor to the podium. This is without a doubt a once-in-a-lifetime opportunity and a sincere privilege. As Dr. Moto mentioned earlier, your work has saved millions of lives and has helped improve the quality of life of so many more than that. Your discoveries are some of the most influential and important that our generation will ever see. Your presence here is really inspiring personally to me. Because I too dream of being involved in biomedical research and would love to accomplish something such as you have in your career. I would live a happy life if I were able to improve even the quality of life for half as many people as you have, and it's a sincere privilege.
As young scientists, Luc Montagnier's hard work and success has inspired us to believe that any goal is attainable with enough hard work and dedication. Aside from setting an example as an impeccably dedicated researcher, your leadership and collaborative spirit inspire scientists around the world to work together to better the quality of life for mankind. Because of the discoveries you have made, our generation has much more hope that AIDS can potentially be effectively prevented and managed. I know that I can speak on behalf of the entire student body when I say thank you for honoring us with your attendance and would like to present you with a small gift of appreciation.
Thank you.
We would like to encourage you to open the gift now, please.
So there is still room for many new discoveries, and I count on you.
Professor, we would actually be very honored if you would open the gift. Sorry, could you please open your gift?
Oh, sorry.
So I hope that you are as thrilled to accept these gifts as we are to present them to you. It is our hope that you will It is our hope that you will be the envy of your colleagues when they see your intimidating Fear the Poet hat. Because, as we know, nothing instills fear quite as well as the Whittier College poet. On behalf of Whittier College, we would like to thank you again for this privileged visit. Let's please get a huge round of applause for Professor Luc Montagnier.
Thank you so much, all of you, and thank you all for coming to this inaugural event of the Center for Science, Health, and Policy. This center is primed to create the kind of interdisciplinary experience and research and policy development that we have celebrated today. And so it was wonderful to have you as part of this wonderful day. Whittier College has a proud tradition of educating scientists and health professionals and teachers of science who rise to the tops of their fields because of the broad education that they received here. Professor Montagnier, you became a poet, and therefore you earned that hat, after a quite illustrious career and after earning the Nobel Prize. One day, I am sure, we will be honoring another Nobel laureate who was first a Whittier College poet and then went on to an illustrious career and a Nobel. I know that such a person is out there, and it's very clear that you inspired them today. We thank you again, and thank you all for coming. Thank you.